For this segment of the lecture, we'll be discussing on how DNA is bridged into proteins. And what bridges DNA to proteins are the ribonucleic acids or RNA, specifically the mRNA or the messenger RNA. Now, a brief review of the central dogma of molecular biology. Central dogma of molecular biology states that DNA makes RNA and RNA makes protein. The overall process of formation of proteins will involve the transcription of DNA to form RNA and the translation of RNA to form protein. Now, the process of gene expression for eukaryotics and prokaryotic organisms may somewhat differ. It's because in the prokaryotic organism, you don't have any nucleus or nuclear membrane that would separate the genetic material from the cytoplasm. In the eukaryotic cell, you have a nucleus providing a separate compartment for these processes. So in a bacterial cell, mRNA is produced from transcription. Immediately, that mRNA is translated without further processing. For eukaryotic cell, the presence of the nucleus provides a separate compartment, and the original RNA transcript called pre-mRNA is processed before leaving the nucleus as mRNA. So in the process of transcription in eukaryotes, the initial product is what we call the pre-mRNA. And that pre-mRNA will undergo several processes of RNA processing to make that mRNA to be utilized for translation. Transcription is the synthesis of RNA using information in the DNA. It is rewritten from DNA to RNA and forms the messenger RNA or mRNA. Translation is the synthesis of a polypeptide using the formation in the mRNA, and translation is also known as protein synthesis. So now let's have a deeper discussion on RNA synthesis called transcription. The term gene has many definitions. For this class, the definition of a gene is a segment of DNA used to make a product that plays a functional role in the cell, maybe either an RNA or a polypeptide. And transcription is the first step in gene expression. It's not replication because gene expression would involve the reading of the DNA segment and not the replication. Okay. Let's have some words to know again. Transcription is the act or process of making a copy. Example of which is when a court reporter hears the witness speaking in English and types a written copy in English of the witness statement. So that is a transcriber. That's the work of a transcriber. He or she will type whatever a witness will tell in the court and type it down to form a written copy. Translation expresses the meaning of words or text in another language, and dogma is a set of principles laid down by an authority as incontrovertibly true. Now, in genetics, the term transcription refers to the copying of a DNA sequence into an RNA sequence. Now, in transcription, it is not the entire DNA sequence that is copied, only a segment of DNA, and that segment is what we call the gene. And only one strand is copied. The structure of DNA is not altered as a result of this process, and it continues to store information and can be transcribed again and again and again. So if that segment of the gene has already been transcribed, it can be transcribed again. Structural genes encode the amino acid sequence of a polypeptide. It's not just example of structural genes. You have collagen, you have elastin, you also have fibrillin. Transcription of a structural gene produces messenger RNA called mRNA. The mRNA nucleotide sequence determines the amino acid sequence of a polypeptide during translation. The synthesis of functional proteins determines an organism's trait. And this path from gene to trait is called the central dogma of genetics or central dogma of molecular biology. So just to reiterate the importance of knowing the central dogma, I am intentionally repeating the process of central dogma because I want to stick it into your mind, these processes, because this is the fundamental basis of life. Okay? For it to be called gene expression, you have to take out DNA replication. Gene expression only involves transcription and translation and whatever processes are present in between. So this is the overall process by which the information within a gene is used to produce a functional product, which can, in concert with environmental factors, determine a trait. The stages of transcription occurs in three parts or in three stages. We have initiation, elongation, and termination. These steps involve protein-DNA interactions. Proteins such as RNA polymerase interacts with DNA sequences. So we have a lot of similarities of the processes of DNA replication and RNA synthesis, or DNA synthesis and RNA synthesis, or transcription. Okay, just to clarify things out, when you say transcription, it is also known as RNA synthesis. When you say replication, it is also known as DNA synthesis. Therefore, you can actually compare and contrast replication from transcription, and even replication from transcription from translation. So hopefully by the end of this lecture, you can be able to find the similarities and differences of these processes. The process of transcription involves initiation, elongation, or what we call the synthesis of RNA transcript, and termination. But this is the overall process. Okay? So in the process of initiation, an RNA polymerase binds to a promoter. Okay? We have a promoter region of a certain segment of DNA. Now, in a gene, we have this called a transcription unit. Okay? And upstream of that transcription unit is a promoter. Okay? Later on, I will be discussing the nitty-gritty of the details of each one, but I'm just going to give you an overview or a bird's eye view on the transcription process. So in transcription, RNA binds to a promoter, okay? And after it binds to the promoter, the DNA strands unwind, just like the helicase. And the polymerase initiates RNA synthesis on the start point on the template strand. So the elongation process would involve the use also of the RNA polymerase. So the RNA polymerase moves downstream, unwinding the DNA and elongating the RNA transcript in 5' to 3' in direction. In the wake of transcription, DNA strands reform a double helix. And the last process is termination. In termination, the RNA transcript is released and the polymerase detaches from the DNA. So this is the RNA polymerase. Once the RNA transcript has already been formed, will be released from the polymerase, and then the DNA will recover or will form wounding again. Okay. So in eukaryotic transcription, we have also a eukaryotic promoter, just like in prokaryotic, and it commonly includes a theta box, or not. This is a TATA box, which is a nucleotide sequence containing thymine and adenine, about 25 nucleotides upstream from the transcriptional start point, or from the point where the RNA polymerase will copy to form mRNA. So in eukaryotic cells, proteins called transcription factors mediate the initiation of transcription by RNA polymerase II. In the second step, several transcription factors recognizes the theta box and must bind to the DNA before RNA polymerase II can bind to the correct position and orientation. For the third step, additional transcription factors, such as these blue objects, bind to the DNA along with the RNA polymerase II, forming the initiation complex or the transcription initiation complex. RNA polymerase II then unwinds the DNA double helix and RNA synthesis begins at the start point on the template strand. For transcription elongation, RNA polymerase moves along the DNA template strands, joining complementary RNA nucleotides to the 3' end of the growing RNA transcript. So the 3' end is quite familiar to you because it's similar to the DNA replication or DNA synthesis, wherein nucleotides are joined at the 3' end. And the synthesis runs from 5' to 3' in direction. So that's one similarity with DNA replication. So that is also true for RNA synthesis. The addition of nucleotide occurs at the 3' end. And the mechanism of termination differs between bacteria and eukaryotes. In bacteria, transcription proceeds through a terminator sequence in the DNA. And transcribe termination function as a termination signal, causing the polymerase to detach from the DNA and releases the transcript. It requires no further modification before translation. The product, the mRNA product, does not undergo any modification. It doesn't undergo any changes. It is what it is in bacteria. In eukaryotic cells, the initial product is what we call the pre-mRNA or the pre-messenger RNA. And this pre-mRNA, and that is unlike in bacteria or prokaryotes, it undergoes alteration of mRNA ends in RNA splicing, which we'll be talking about soon. In RNA processing, we have processes like addition of the 5' cap and the poly A tail. And splicing involves the removal of the introns, leaving only the exons. So later we'll be discussing what are these introns and exons. So basically exons, they are the coding regions of the segment of a gene, while introns are the non-coding regions of a gene. Again, when you say exon, these are coding regions, meaning they are the specific sites or specific DNA segments where your ribosomes will read on. Now, the introns are non-coding region. RNA transcripts have different functions. Once they are made, RNA transcripts play different functional role. Over 90% of all genes are structural genes, which are eventually transcribed to mRNA. The final functional products are polypeptide. Other RNA molecules in this table, you see here later, are never translated. It's only the mRNA that is translated. The final products are RNA molecules in the process of transcription. The RNA transcript from non-structural genes are not translated. They do have various important cellular functions, and they can still confer traits. In some cases, the RNA transcript becomes part of a complex that contains protein subunit, example of which are ribosomes, spliceosomes, and signal recognition particles. Okay? Now, not all RNA will be read by the ribosome. That's the main point of this slide. It can be a part of the ribosome, of a spliceosome, and some signal recognition particles. So as you can see, this table shows you the different functions of the RNA molecules. So you have this column shows you the type of RNA and their description. It's nice to know for you to be familiar with the functions of the RNA molecules, because we are now in the era of molecular research, and if you are planning to have a research in the medical science in the future, you should be able to know the functions of RNA, the different types of RNA, because that's one way of this. That's an idea for your part to venture in transcriptomics. That's a new field in the biological science. When you say transcriptomics, we discuss or we study about the processes that occur during the transcription process. So we have the various types of RNA. We have the mRNA, tRNA, ribosomal RNA, microRNA. We have the small cytoplasmic RNA, RNA-Sp, small nuclear RNA, telomerase RNA, small nucleolar RNA, and the viral RNAs. Our molecular understanding of gene transcription came from studies involving bacteria and bacteriophages. Indeed, much of our knowledge comes from the study of a single bacterium, which is the E. coli. Now, just like in DNA replication, um, our model for the understanding of the replication process is E. coli. Now, in transcription, it's also the E. coli. So in this section, we will examine the 3 steps of transcription as they occur in bacteria. Okay, so we need to know the general process of transcription in bacteria because it's quite simpler compared to the eukaryotes. Later, we'll be discussing about eukaryotes. But before we start our discussion on the specific steps of RNA synthesis or transcription, we need to define some important terminologies that are involved in the processes. When you say promoters, so these are DNA sequences that promote gene expression. More precisely, they direct the exact location for the initiation of transcription. It tells our cell that, hey, this is, or it tells our RNA polymerase that, hey, this is the site of gene X. For example, gene X codes for collagen. So this is the site. So dito tayo mag-start ha. So dito ang starting point mo, dito ka mag-bind. So promoters are typically located just upstream of the site where transcription of a gene actually begins. Now, a promoter site, that site is not actually transcribed. It's just a binding site for that RNA polymerase. The bases in a promoter sequence are then numbered in relation to the transcription start site. Okay. What are the common features of promoters in E. coli? There are 2 consensus sequences at -10 and -35 for the subunit binding for the RNA polymerase. And this is what we call the theta sequences, okay? Or the tata sequences. When you see -10 and -35, it refers to 10 nucleotides away or 10 nucleotides before the start of the transcription site, or the transcription start site. Okay, so when you see -10, it refers to 10 nucleotides before the transcription start site, and we also have -35, which is 35 sequences before the start site. And you see this Latin sign. This is a sigma subunit binding. This is a portion of the RNA polymerase that specifically binds to the promoter. It is a segment or a part of the RNA polymerase that binds to the promoter to help direct RNA polymerase to bind to that site. An AT-rich upstream promoter element between -40 and -60 binds the alpha subunit of the RNA polymerase. An AT-rich sequence promotes strand separation. And these sequences govern efficacy of RNA polymerase binding and therefore affect gene expression level. Nucleotides before the first nucleotide of the RNA molecule are considered upstream and given negative value. That is why it is a -35, it's -10. Okay? So as you can see here, this is the usual structure of a promoter sequence. So this is the start of RNA. This is the start of RNA, of the transcription, where the RNA polymerase will really start the copy of the DNA. Now, before that, the nucleotides before the transcription start site, those values are negative. So these are examples of E. coli promoter regions. We have the -10 region and -35 region. As you can see here, these are different genes. So you have RRNBP1 gene, you have the TRP gene, the LAC gene, the REC-A gene, and AROBAT gene. So you see this region, this purple region, these are the promoters. And it is in this part where the subunits for the RNA polymerase will bind to, because it will help the RNA polymerase guide that enzyme on which genes to be transcribed. Okay. So again, This is the transcription start site. Most of the promoter region is labeled with negative values. It's because it is from negative 10 amino acid, no, negative 10 nucleotide sequence from the transcription start site, and you have negative 35. It refers to a negative 35 nucleotides away from the transcription start site. So these are the promoter regions. We have a negative 10 sequence and negative 35 sequence. The negative 10 sequence consists of the TATA, while negative 35 you have TTGA, CA. The sequence element that play are the sequence elements. These play a key role in transcription. So this negative 10 sequence is also known as the Pribno box. Okay, the negative 10 sequence promoter is also known as the Pribno box. Again, what's the importance of knowing this? It is where a segment or a subunit of RNA polymerase will bind to help initiate transcription. So the initiation of bacterial transcription involves RNA polymerase. And RNA polymerase is the enzyme that catalyzes the synthesis of RNA. Now in E. coli, RNA polymerase holoenzyme is composed of a core enzyme and a sigma factor. The core enzyme consists of 5 subunits, and the sigma factor contains 1 subunit. And these subunits play distinct functional roles. Again, it is a holoenzyme, meaning consists of several subunits. It has 5 core subunits of this one, and plus a 6 called the sigma. So this is 26.4. Okay, what is important about RNA polymerase is the sigma subunit. The sigma subunit, it is the one that, this is the sigma factor, the sigma subunit. It is the one that binds to this portion, to the promoter. Okay. Now, what is also unique about RNA polymerase is that it lacks a 3' to 5' exonuclease activity, unlike that in DNA polymerases. So it has a high error rate. And RNA polymerase binds to promoter regions to initiate transcription. Okay. 2 alpha units function in the assembly and binding to the upstream or promoter element. The beta subunit is the main catalytic subunit. So it's the beta subunit. It's the beta subunit that is the one that is carrying out the actual process of making the RNA or polymerizing RNA. The beta prime subunit is responsible for DNA binding. The sigma subunit directs the enzyme to the promoter. And each class of RNA polymerase holoenzymes has a different sigma subunit. So again, the importance of the sigma subunit is that it directs the enzyme to the promoter site. Again, the promoter site is the upstream of the transcription start site. And this symbol, this like it press W, this is omega. The omega subunit appears to protect the polymerase from denaturation. So what happens during initiation? The RNA polymerase holoenzymes bind loosely to the DNA. It then scans along the DNA until it encounters a promoter region. And when it does, the sigma factor recognizes both the negative 35 and negative 10 region. Okay, this is a region within the sigma factor. This sigma factor contains a helix-turn-helix structure which is involved in tighter binding to DNA. Okay, so the alpha helices binding to major groove. These are the components of the sigma factor. So the sigma factor has an a helix-turn-helix structure. A helix-turn-helix structure. So it's the sigma factor that specifically binds to the promoter region. The binding of the RNA polymerase to the promoter forms the closed complex. Then the open complex is formed when the TAT box in the negative 10 region is unwound. Again, the other name for the negative 10 region is the Pribno box. A short RNA strand is made with the open complex, and the sigma factor is released at this point. and marks the end of initiation. The core enzyme now slides down the DNA to synthesize an RNA strand, and this is known as the elongation phase. Okay, so again, binding of RNA polymerase, the promoter, specifically the sigma factor, forms the closed complex. Okay, when the TATA box or the Pribnow box is opened, okay, in the -10 region is open or unwound or separated, you've now formed the open complex. Now, the RNA polymerase will try to read that portion. A short RNA strand is made with the open complex. And once this strand is formed, it now releases the sigma factor. Okay? So this is the RNA polymerase. It binds to the promoter region with the help of the sigma factor. Now, after sliding along the DNA, the sigma factor recognizes a promoter, and RNA polymerase polyenzyme forms a closed complex. So this is the closed complex. An open complex is formed and the short RNA is made. Okay? When a short RNA is made, the sigma factor is then released and the core enzyme is able to proceed down the RNA. Now, the trigger for the elongation process is the release of the sigma factor. Okay? And why does the sigma factor will be released? Is because of a short RNA is synthesized. So that is the triggering point when the sigma factor will be released. And if the sigma factor is released, the core enzyme, the RNA polymerase core enzyme, will now proceed its process of transcription or RNA synthesis. The RNA transcript is synthesized during the elongation stage. Okay. Now, in the initiation phase, there is no RNA transcript yet that is formed. Only a short segment of that of an RNA is only formed to release that sigma factor. The DNA strand used as a template for RNA synthesis is termed the template strand. Again, the DNA strand used as a template for the RNA synthesis is termed as the template strand. The opposite DNA strand is called the coding strand. So let's see that one later. So in transcription, RNA polymerase reads only one strand of the DNA, and it reads the template strand. I am emphasizing RNA polymerase reads one strand of DNA. And that portion of DNA where the RNA polymerase is read is what we call the template strand. It moves along the template strand from 3' to 5'. Okay? Are you familiar with this one? It reads the DNA from 3' to 5'. This is also similar to the process of DNA replication. The RNA polymerase simultaneously makes an RNA copy of the template strand's complementary partner, and the partner strand is called the coding strand. The new mRNA molecule is made in 5' to 3' in direction, again, similar to DNA replication. The orientation and sequence of mRNA is identical to the coding strand of the DNA, except use for these. Again, to further clarify which is a template, which is a coding strand. This is the DNA. DNA is double-stranded, right? And our RNA is single-stranded. The portion where the RNA polymerase will read the DNA is what we call the template strand. And the template strand runs from 3' to 5'. Opposite to that, or complementary to the template strand, is what we call the DNA non-template or the coding strand. And the product of RNA synthesis is what we call the RNA transcript. So when we refer it to coding strand, when you hear the term coding strand, we are referring to the DNA, not the RNA. When we refer to the template strand, we are referring to the DNA where the RNA polymerase will read to form that RNA transcript. So for this one, which is the template strand, which is the coding strand? The template strand for this one is this one. The coding strand is this one. Why is it called the coding strand? Because the coding strand will have a similar base sequence as that of the RNA transcript. The open complex formed by the action of RNA polymerase is about 17 bases long. Behind the complex, the DNA rewinds back into a double helix. On average, the rate of RNA synthesis is about 43 nucleotides per second. Okay. So key points. RNA polymerase slides along the DNA, creating an open complex as it moves. The DNA strand, also known as the template strand, is used to make a complementary copy of RNA as an RNA-DNA hybrid. So this is the RNA-DNA hybrid. So RNA polymerase it is the one is responsible for the unwinding of the DNA. Unlike in DNA replication, you need to have a separate enzyme for that, separate protein called the helicase. For this one, you don't have any helicase. It's actually the RNA polymerase that acts like a helicase at the same time, a polymerase. So RNA polymerase moves along the template strand in a 3' to 5' direction, and RNA is synthesized in 5' to 3' direction using nucleoside triphosphate as precursor, and pyrophosphate is released. And this process is very, very similar to the synthesis of DNA via DNA polymerase. The complementary rule is the same as the ATGC rule, except U is substituted for T in the RNA. The last step for bacterial transcription is termination. Termination is the end of RNA synthesis. It occurs when the short RNA-DNA hybrid of the open complex is forced to separate. This releases newly made RNA as well as the RNA polymerase. It's the end. It is the final step of transcription. And E. coli has two different mechanisms for termination. We have a row-dependent termination and row-independent termination. For row-dependent termination, it requires a protein known as Rho. So this symbol, like a P, but this is actually Rho. For row-independent, it does not require Rho. So what is this row-dependent and row-independent termination? So the first type of termination in E. coli is the row-independent termination. It is characterized by 3 uracil nucleotides near the 3' end of the transcript. Okay. Self-complementary regions in transcript forms a hairpin of 15 to 20 nucleotides before the 3' end, and this causes the RNA polymerase to pause and causing the dissociation. So for example, in this illustration, in a row-independent termination, the RNA polymerase will read. Now, when an RNA polymerase forms a transcript made up of 3 uracil residues or 3 uracil nucleotides in the transcript, now forms this hairpin loop. Right? This RNA hairpin is formed at a palindromic sequence, reducing the length of the RNA-DNA hybrid. So this loop causes a destabilization of this complex. So what happens is that the mRNA is released and the polymerase is dissociated. So that is the row independent, meaning there's no involvement of a row protein. It's just that a transcript of 3 uracil nucleotides are read and forms a hairpin loop, causing the dissociation of the RNA polymerase. For row-dependent termination, okay, a common CA-rich sequence called a root site, or the root site stands for the row utilization element, is usually present. The row protein processes until termination site is reached, and row protein is actually quite similar to a helicase and binds to the rat site and causing the dissociation of mRNA. So in row-dependent termination, the row helicase binds to a rat site of that is found in the transcript. So it's in the transcript. Okay. So that row protein will migrate along the mRNA to the elongating RNA polymerase. So it follows through the RNA polymerase. Okay? And the raw helicase separates the mRNA from the DNA template, causing the release of the mRNA and subsequent release of the RNA polymerase. Okay. So again, another illustration, raw protein is a helicase. So when a raw utilization element is transcribed, it attracts now the raw protein. Now that raw protein is a helicase. Okay, remember the raw protein is a helicase and moves towards the 3' end of the transcript. The RNA polymerase reaches the terminator. A stem loop causes RNA polymerase to pause. Okay, that stem loop here, that's just like similar to in raw dependent, forms like a hairpin loop. The RNA polymerase pauses due to the interaction with the stem loop structure, and raw protein catches up to the open complex and separates the RNA and DNA hybrid. Okay, so remember that raw is a helicase, so it separates now whatever wounded nucleic acids. So if there's a DNA or RNA hybrid, remember that the DNA and RNA hybrid forms a double helix, it forms a wounding or binding together. So that raw helicase will separate or will unwound this hybrid to release that mRNA. Again, to emphasize, for raw independent termination, it is facilitated by two sequences in the mRNA. A uracil-rich sequence made up of 3 uracil nucleotides found at the 3' end of the RNA, and a stem loop structure, or what they call the hairpin loop structure of the uracil-rich sequence, causes the termination of the RNA synthesis. Now let's move on to the transcription in eukaryotes. Many of the basic features of gene transcription are very similar to bacteria and eukaryotes. However, gene transcription in eukaryotes is more complex. A larger, more complex cells or organelles are usually involved, and added cellular complexity means more genes that encode proteins are required. This multicellularity adds another level of regulation, and they express genes only in the correct cells at the proper time. Okay. So in eukaryotic RNA polymerases, it is transcribed, or the nuclear DNA is transcribed by 3 different RNA polymerases. We have RNA Pol 1, RNA Pol 2, and 3. RNA polymerase 1 transcribes all ribosomal RNA genes except for the 5S ribosomal RNA. RNA polymerase 2 transcribes all structural genes, thus synthesizes all the mRNAs, and it transcribes some snRNA or small nuclear RNA genes. For RNA polymerase 3, it transcribes all the tRNA gene and the 5S ribosomal RNA gene. So the RNA polymerase 3 is responsible for formation of the tRNA, or transfer RNA. Eukaryotic promoter sequences are more variable and more often complex in eukaryotes than those in bacteria. For structural genes, at least 3 features are found in most promoters. We have a regulatory element, we have a data box, and the transcription start site. The core promoter is relatively short. It consists of the data box and the transcription start site, and it is important in determining the precise start point for transcription. The core promoter by itself produces a low level of transcription, and this is what we call basal transcription. So the promoter itself undergoes this process called basal transcription, and the transcription start site is usually an adenine. Regulatory elements are short DNA sequences that affect binding of RNA polymerase to the promoter. So this is in relation to eukaryotic Transcription factors or proteins bind to these elements and influence the rate of transcription. Okay. Now, I would like to emphasize that promoter is different from regulatory elements. Usually, promoters are closer to the actual site of transcription, and regulatory elements are usually upstream, okay? Upstream from the transcription start site. The transcription factors, these are proteins that binds to these elements, to these regulatory elements, and they influence the rate of transcription. So this is the reason why our cells of our body have a different genes that are expressed because of the binding of several transcription factors to these regulatory elements. So not all genes are transcribed in a certain cell. Depends on the function of the cell. It depends on the transcription factors that are found within that cell. And there are two types of regulatory elements. We have enhancers and silencers. Enhancers, they are regulatory elements wherein if a transcription factor is bound to, it stimulates transcription. When you say silencers, these are segments or regulatory elements wherein if the transcription factor is bound to that, it inhibits transcription. Okay? The regulatory elements vary widely in their location but are often found in the -50 to -10 region. So it's really upstream away from the transcription start site. So factors that control gene expression can be divided into two types, and it's based on their location. When you say cis-acting elements, these are DNA sequences that exert their effect only over a particular gene. Example of which are the data box, the enhancers, and silencers. When you say trans-acting elements, these are regulatory proteins that bind to such sequences. So cis is the DNA sequence, while trans is the protein that binds to these sequences. RNA polymerase II and its transcription factors. There are three categories of proteins that are required for basal transcription to occur at the promoter. Okay, so we have three categories of proteins required for the transcription in eukaryotes. You have the RNA polymerase II. You have 5 different proteins called general transcription factors or GTFs, and a protein complex called the mediator. We won't go over this. So figure 12.14 shows the assembly of the transcription factors and RNA polymerase II at the data box. Okay. So in this process, so it shows you the assembly, it's basically the assembly of transcription factors at the data box. It's very complicated in the whole process. The main point here is we utilize a lot of transcription factors to the promoter side. Okay, so in this illustration, you see here that the TF2D binds to the data box. In prokaryotics, it's actually the sigma factor that binds to the data box. And TF2D is a complex of proteins that includes the Tata binding protein or TBP and several TPB associating factors or TAFs. Now, once TF2D is bound to the Tata box or the data box, TF2B binds to TF2D. Now, TF2B acts as a bridge to bind RNA polymerase II and TF2F. Okay? So TF2B forms a bridge of TF2F and the RNA polymerase II. TF2E and TF2H bind to RNA polymerase II to form a pre-initiation or closed complex. So this is the pre-initiation complex or closed complex made up of the TF2B, TF2B, TF2F, TF2H, TF2E, and the RNA polymerase. The TF2H acts as a helicase to form an open complex. So remember the action of the helicase in DNA synthesis, it unwounds the DNA. TF2H also phosphorylates the CTD domain of RNA polymerase II. CTD phosphorylation breaks the contact between TF2B and RNA polymerase II. So again, what's the function of TF2B? It acts as a bridge between RNA polymerase II and the TF2D. So once TF2, once the CTD is phosphorylated. It breaks the contact between TF2B and Rna polymerase 2. TF2B, TF2E, and TF2H are then released. Okay, and once this happened, the release, the release of the TF2B, TF2E, and TF2H causes now the elongation stage. So it marks the elongation stage. So this is how complicated the eukaryotic transcription process is. Compared it to the prokaryotic. So how are the RNA polymerases in eukaryotics happen? So the pre-mRNAs are modified by decleavage near the 3' end with subsequent attachment of a string of adenines. And transcription terminates 500 to 200 nucleotides downstream from the poly A signal. There are two models for termination, but we won't be covering this much because it's a complicated one. Now let's move on to RNA modification. Now, once RNA is already formed from the process of RNA synthesis by the RNA polymerase, okay, some of these RNAs, all of the RNAs that are produced in eukaryotes undergo some series of RNA modification. So that RNA will not be immediately read by the ribosome. It needs to go some processing. And that is what we call post-transcriptional modification. RNA modification is also known as post-transcriptional modification. Analysis of bacterial genes in 1960s and 1970s revealed the following. The sequences of DNA in the coding strand corresponds to the sequence of nucleotides in the mRNA. So this is for the bacteria. The sequence of codons in mRNA provides the instructions for the sequence of amino acids in the peptide. And this is termed collinearity of gene expression. So there is a collinearity of gene expression for bacterial genes because the sequence of DNA is actually the same as that of the mRNA that is produced. Analysis of eukaryotic structural genes in the late 1970s revealed that they are not always collinear with their functional mRNAs. So this has led to the idea that RNAs or mRNAs that are produced after transcription are really modified. So we have coding sequence called exons. These are interrupted by intervening sequence called introns. Transcription produced the entire gene product. Introns are later removed or excised, and exons are connected together or spliced. And this phenomenon is termed RNA splicing. It is a very common genetic phenomenon in eukaryotes, but some bacteria also follow RNA splicing. But when you hear of RNA splicing, it's usually in eukaryotes. So when you say exons, these are coding sequence. It is actually the sequences that are read by the ribosomes to form the polypeptide sequence. And the sequences between the exons, they are not read by the ribosome. Therefore, they have to be removed before the ribosomes will read the mRNA transcript. So introns are later removed through the process of excision, and the connection of several exons together, and that is what we call splicing. So aside from splicing, RNA transcript can be modified in several ways. For example, from trimming of ribosomal RNA and tRNA transcript, and the 5' capping and the 3' poly-A tailing of mRNA transcript. So we have a lot of RNA modification, of post-transcriptional modification. So try to focus your attention here. So modification can occur in RNA, can be through degradation of RNA, can be through splicing, 5' capping, poly-A tailing, RNA editing, or base modification. But these three are most commonly found in eukaryotes. studied in eukaryotes. So we have splicing, the 5' capping, and the 3' poly A tailing. For splicing, we have 3 different splicing mechanisms that have been identified. We have the group 1 intron splicing, group 2 intron splicing, and the spliceosome. We will focus on spliceosome. All of these 3 cases involve the removal of the intron RNA and the linkage of the exon RNA by a phosphodiester bond. So we have different types of intron splicing. So if you have a group 1 intron, the mechanism of removal is self-splicing. For group 2 is through self-splicing as well. For pre-mRNA, it's through a spliceosome. It's very commonly found in structural genes within the nucleus of the eukaryotes. So let's discuss about spliceosome because it's one of the most commonly found in structural genes in eukaryotes. So in eukaryotes, the transcription of structural genes produce a long transcript known as a pre-mRNA. So once the RNA polymerase 2 of eukaryotes reads the DNA template from 3' to 5', it now produces this RNA called pre-mRNA. This RNA is altered by splicing and other modification before it leaves the nucleus. Okay? Splicing in this case requires the aid of a multi-component structure known as a spliceosome. So the spliceosome is a large complex that splices the pre-mRNA. It is composed of several subunits known as the SNRPs, or the small nuclear ribonuclear proteins, or ribonuclear RNA, and a set of proteins. So it is called a SNRPs. Okay, so this is the whole process of general process of the spliceosome. So the introns, we want to get rid of the introns before it is read by the ribosome, before it leaves the nucleus. Because again, the exon is the one that we want. That is the coding region. That is what the ribosomes will read later in the translation process. So since you still have introns, you need to remove the introns using the spliceosome. So with this, we will remove the intron, and the remaining exons, we will try to glue them together to form this mature mRNA that can eventually leave the cell that is ready to be read by the ribosome. The subunits of a spliceosome carry out several functions. First, it binds to an intron or an intron sequence and precisely recognizes the intron exon boundaries. Second, it holds the pre-mRNA in the correct configuration. And third, it catalyzes the chemical reaction that removes introns and covalently linked exons. The intron RNA is defined by a particular sequences within the intron, not the intron exon boundaries. So our spliceosomes are very smart also because they can recognize which is an intron. Okay? So the consensus sequence for this for the splicing of mammalian pre-mRNA is shown here. So I don't expect you to memorize them. My main thought for this one, my main point, is that there are some sequences in the introns where the spliceosome can detect to recognize as an intron, so that we can remove this intron. So in the process of pre-mRNA splicing using the spliceosome, okay, we have this 5' splice site and this 3' splice site. In the process, U1 of the spliceosome, this is actually a subunit of a spliceosome. U1 SNRPs binds to the 5' splice site. U2 binds to the branch site. Okay? And the U4, U6, and U5 trimers bind after the binding of the U2 SNRPs. And intron loops out of the exons and are brought closer together. So as you can see here, it brings this intron sequence close together. So that's the main point why there's a branch site, where there's a U2 site, or there's a U2 SNRP, for it to form this loop arrangement. So this intron intron loops. It loops out and exons are brought closer together. So the importance of forming this loop structure is to bring closer the exons because we want to combine these two exons. The 5' slice is cut. So this is the 5'. So the 5' slice is cut and the 5' of intron is connected to the A in the branch site to form a lariat. Okay, so I forgot to mention about the A site. So the A site is part of a specific part of a branch site because that is also important because the 5' site of the intron binds to it to form a lariat structure. So the 5' slice is cut and the 5' end of the intron, so this is the intron, the green one. So the 5' is cut. And now that 5' end binds to the A in the branch site to form a lariat. This is the structure of a lariat. And the U1 and U4 are released. And now the 3' slice is cut. So you cut through here. And exon 1 is connected to exon 2. And the intron in the form of lariat is released together with the U2, U5, and U6. And the intron will be eventually be degraded. Okay? So as you can see here, the exon 1 and exon 2 is now connected. So again, the main point of doing this configuration is just to bring the two exons together. And this portion is the introns. You want to get rid of the introns because you will not be using this one for the translation. So we need to bring the exon 1 and exon 2 close together to form this loop and eventually this configuration forming a lariat. This lariat is formed because of the 5' end of that intron after that is being cut from the exon 1 attaches to the A site of the branch site. What is the advantage of having introns? One benefit of genes with introns is a phenomenon called alternative splicing. A pre-mRNA with multiple introns can be spliced in different ways. This will generate mature mRNAs with different combinations of exons. And this variation in splicing can occur in different cell types or during different stages of development. The biological advantage of alternative splicing is that 2 or more polypeptides can be derived from a single gene. And this allows an organism to carry fewer genes in its genome. One very important biological advantage again is in the eukaryotes is the phenomenon of alternative splicing. Okay, so the degree of splicing and alternative splicing varies greatly among different species. For a baker's yeast, also known as the Saccharomyces cerevisiae yeast, contains around 6,300 genes. And around 300 of that genes encode mRNA that are spliced. And only few of these 300 have been shown to be alternatively spliced. Human contains around 25,000 genes. Most of these encode mRNA that are spliced. And it is estimated that about 70% are alternatively spliced. Now, certain mRNAs can be alternatively spliced to produce dozens of different mRNA. So the next image considered, this image considers an example of alternative splicing for a gene that encodes alpha-tropomyosin. Now, alpha-tropomyosin is a protein that functions in the regulation of cell contraction. It is found in smooth muscle cell, in striated muscle cell, and it's also found in many types of non-muscle cells at low levels. The different cells of multicellular organism regulate contractibility in subtly different ways. So one way to accomplish this is to produce different forms of alpha-tropomyosin by alternative splicing. So alternative splicing will create diversity in the production of mRNA. So as you can see here, you have the exons. The colored one are exons. The line ones, these are the introns. So in alternative splicing, we have various ways of connecting the different the exons. So these alternatively spliced version of tropomyosin vary in function to meet the needs of the cell type in which they are found. So In the main point of alternative splicing is to create diversity of the transcript. Alternative ways of the rat alpha-tropomyosin pre-mRNA can be spliced. So this is the overall process. Most mature mRNA have a 7-methylguanosine covalently attached to their 5' end, and this event is known as capping. Capping occurs as the pre-mRNA is being synthesized by RNA polymerase II. It is usually when the transcript is only 20 to 25 bases long. So as shown here, capping is a three-way process. So you have now the transcript, the mRNA transcript. At the 5' end, you now have the phosphate group. So this phosphate group will be the substrate for the RNA 5' triphosphatase, which removes a phosphate, an extra phosphate from the 5' end. Okay. Guanylyl transferase hydrolyzes GTP, okay, GTP to form GMP. The GMP is attached to the 5' end, okay, and pyrophosphate is released. Okay, so you have the first enzyme, you have the RNA 5' triphosphatase. It removes the phosphate, one phosphate at the 5' end. And using this enzyme, guanylyl transferase, it hydrolyzes GTP to form GMP. So now, GTP to form GMP, you release two phosphate group called pyrophosphate. And the resulting GMP will now bind to the phosphate group. Then methyltransferases attaches a methyl group to the guanosine. It attaches an extra methyl group to the guanosine. Okay, and this is what we call the 7-methylguanosine cap. So the 7-methylguanosine cap structure is recognized by cap binding proteins. So these cap binding proteins play a role in the movement of some RNA into the cytoplasm, the early stages of translation, and also in the splicing of introns. Most mature mRNAs have a string of adenine nucleotides at their 3' end. So this is at the opposite end. And this is termed as the poly-A tail. So it consists of adenine nucleotides. The poly-A tail is not encoded in the gene sequence, and it is added enzymatically after the gene is completely transcribed. So this is what we call the polyadenylation. Okay, so this is a polyadenylation. There is a consensus sequence in higher eukaryotes once an mRNA is formed. The endonuclease cleavage occurs about 20 nucleotides downstream from this sequence. Okay, an endonuclease, it cleaves a portion of that mRNA. And we have this enzyme called poly-A polymerase. It adds adenine nucleotides to the 3' end. First, an endonuclease cuts that portion when it recognizes this polyadenylation signal sequence. An endonuclease will cut at the lower portion or at the 3' portion, and the poly-A polymerase adds adenine nucleotides to the 3' end. So a lot of adenine nucleotides at the 3' portion. Okay, and what is the function of that poly-A tail? That poly-A tail appears to be important in the stability of mRNA and also in the translation of the polypeptide. Now, the poly-A tail varies between species. It can be a few dozen adenines to several hundred. So that ends the process of transcription. Now let's move on to the final step of the central dogma of molecular biology, which is the translation, or also known as protein synthesis. Translation is the RNA-directed synthesis of a polypeptide. As a molecule of mRNA is moved through a ribosome, codons are translated into amino acids one by one. The interpreters are tRNA molecules, each type with a specific nucleotide triplet called an anticodon at one end and corresponding amino acid at the other end. The tRNA adds its amino acid cargo to a growing polypeptide chain when the anticodon hydrogen bonds to a complementary codon on the mRNA. So what are the key role players in translation process? So here we have the ribosome, we have the mRNA, with the tRNA. The tRNA is bounded to an amino acid, okay? And its product, which is the polypeptide. So what are the features of protein synthesis? Protein synthesis, the process of protein synthesis would entail large energy cost. It can be rapid when accomplished on a cluster of ribosome called a polysome. And in bacteria, translation or protein synthesis is usually coupled to transcription. So in bacteria, translation can begin before translation is finished. And why is that possible? It's because in bacteria, you don't have any compartments, don't have any nucleus. So the ribosomes are readily available to immediately read the mRNA that is produced from transcription. So that is the one advantage of these bacteria or the ribosomes. Okay, so I would just like you to be familiar with the structure of the ribosome. So, and the specific part. So the ribosome is made up of two major subunits. Okay, and it has a larger and a smaller subunit. In the larger subunit, consists of sites such as the A site, the P site, and the E site. Okay, the A site is what we call the amino acid tRNA binding site. The P site is the peptidyl tRNA binding site, and E site is the exit site. And it is in the P site where you have an exit tunnel where your proteins will be released from. So this is the usual process of translation. So later we'll show you the details of the translation process. But before that, let's have a discussion first about codons, which are the triplets of bases. The flow of information from gene to protein is based on a triplet code. So this is a series of non-overlapping 3 nucleotide words. These triplets are the smallest units of uniform length that can code for all the amino acids. During transcription, one of the two DNA strands, called the template strand, provides a template for ordering the sequence of nucleotides in the RNA transcript. During translation, the mRNA-based triplets, called codons, is read in 5' to 3' direction. And each codon specifies the addition of one of the 20 amino acids. So when you hear the word codon, it refers to the triplet of bases found in the mRNA. The genetic code is universal with a few exceptions. It is used by prokaryotes and eukaryotes across species. Mitochondria contains DNA and use a slightly different code. In mitochondria, the UGA encodes for trip in the vertebrate mitochondrial DNA instead of stop. And AGA or AGG encodes stop in the vertebrate mitochondrial DNA instead of arginine. Mitochondria encode their own tRNAs using only 22 instead of 32. Okay, so this is the usual table that you will see when you search for codons, or the what we call the genetic code. So basically what this table shows you is that if your ribosome will read 3 letters of bases consisting of U, it codes for the phenylalanine amino acid. Okay, if it codes for example CCA, if the ribosome reads nucleobases consisting of CCA, it codes for the amino acid proline. If the ribosome will read in the mRNA, if it reads UAA, UAG, or UGA, it codes for a stop, meaning it terminates the process of translation. And all organisms start with AUG. So AUG is the start of translation. It is the codon that is first read by the ribosome, and it is usually the AUG codes for methionine. Wobble base pairing in tRNA with mRNA can occur on the third base. The third base of a codon can form a non-canonical base pairs with its complement. Some tRNA contains inosinate, which can hydrogen bond with a UCNA. Now, these hydrogen bonds are weaker and were named by Crick as wobble base pairs. So, what do you mean by wobble? Okay, so for example, if you have this codon, the third nucleobase has the capacity to be changed, to be varied. Okay, and for example, in yeast, CGA, GCU, and CGC can all bind to tRNA that brings an arginine, which is an anticodon of GCI. Although sequences are usually written in 5' to 3' direction, the anticodon here is written 3' to 5' to illustrate its bonding with the mRNA. So again, a codon will be written from 5' to 3'. So this is the 5' end and this is the 3' end. For the anticodon, it's the 3' end to 5' end because it's the complementary. So how do tRNA recognize the codon by the mRNA? The codon sequence is complementary with the anticodon sequence. The codon in the mRNA base pairs with the anticodon in the mRNA by hydrogen bonding. So the tRNA brings, it has this anticodon and it hydrogen bonds with the mRNA, with the codon. Okay. Again, the codon is found in the mRNA, the anticodon is the tRNA. So the alignment of the two RNA segment is anti-parallel. Okay. So as you can see this table, it shows how the wobble base of the anticodon determines the number of codons a tRNA can recognize. Okay. So we will not deal much about the codons, and I don't expect you to memorize this table because it's a very tedious table. I just want you to remember the stop and the start codons. So the start codon is the AUG and the stop codon is the OWA, UWAG, UGA. So that is the stop codon. The genetic code is resistant to mutation. The generate code allows certain mutation to still code for the same amino acid. And we have that silent mutation, which codes for a different nucleotide in the DNA, but codes for the same amino acid in the protein. Mutation in the first base of a codon usually produces a conservative substitution. Some mRNAs, wait. Now, some mRNAs are edited before protein synthesis. Editing involves alteration, addition, or deletion of nucleotides. That's what we have previously mentioned about transcription or post-transcriptional modification. Before mRNAs are read by the ribosomes, they can be altered, can be modified, or some of the nucleotides are deleted. So, RNA editing by alteration of nucleotides most commonly involves enzymatic deamination of adenosine or cytidine residues to form inosine or uridine. Now let's move on to the stages of protein synthesis. The stages of protein synthesis is classically divided into three, just like in transcription. You have the initiation, elongation, and termination. But there are actually 5 stages of protein synthesis. You have activation of amino acids, the initiation of translation, elongation, termination, and ribosome cycling, and folding and post-translational processing. But the classic stages of protein synthesis involves initiation, elongation, and termination, just like in transcription. So that is a valid similarity between translation and transcription. This is the overview of protein synthesis. The first step is the activation of amino acid. The tRNA is amino acylated, meaning an amino acid will bind to a portion of that tRNA. The second step is initiation. The mRNA and the amino acylated tRNA bind to the small ribosomal subunit, okay, small subunit of the ribosome, and the large subunit binds to it thereafter. Now, elongation is the successive cycles of amino acid tRNA binding and peptide bond formation occur until a ribosome reaches a stop codon. Again, what are the 3 stop codons? We have the UA, UAG, and UGA. UAA, UAG, and UGA. Termination is what happens when translation stops, when a stop codon is encountered, and mRNA and proteins dissociate, and the ribosomal subunits are then recycled. Then after the protein is already been released from the ribosome, it can undergo protein folding and post-translational modification or post-translational processing. Okay, so these are the components for the 5 major stages in protein synthesis in E. coli. We have activation of amino acids. We have, of course, we need the 20 amino acid. We have the 20 amino acid tRNA synthesis, tRNAs, ATP, and magnesium ion. For initiation, of course, you need that mRNA. We have the N-formyl methionine tRNA F-met, which is usually the first amino acid that is usually in place. Initiation codon in mRNA, which is AUG. The 30S ribosomal subunit, the 50S ribosomal subunit. Initiation factors, GTP, and magnesium. For elongation, you have the functional 70S ribosomes form an initiation complex. The amino acid tRNA specified by codons, several elongation factors, GTP, and magnesium. For termination, you need the termination codon in mRNA and some releasing factors, EFG and IF3. For the folding and post-translational processing, you need chaperones and folding enzymes, and enzymes that will add proteolytic processing such as modification of terminal residues of proteins, attachment of acetyl, phosphoryl, methyl, carboxyl carbohydrate, or phosphatic group. But we will only focus ourselves on stage 1 and up to stage 4 because stage 5 is a whole new process and it entails a complicated process. Ribosome is a key player in protein synthesis. And 25% of the dry weight of bacteria is actually ribosomes. 65% of ribosome is made up of ribosomal RNA. So ribosomal RNA is the major composition of ribosome, not protein. It's only 35% protein. RNA does the catalysis of the peptide bond formation. It's actually not the protein. That is why it's called also the ribozyme. Ribosomes are made up of two subunits bound together by 30S and 50S in bacteria, with mRNA running through them. So this is this table shows you the RNA and protein components of the E. coli ribosome. So this is the comparison of the ribosome in the structure in the bacteria. This is the bacteria and the yeast. Ribosomal RNA have a complex secondary structure. The three single-stranded ribosomal RNA of E. coli have a specific 3D structure with extensive interchain base pairs, and the shapes of the ribosomal RNA are highly conserved. So I just want you to be familiar that the ribosomal RNA, they are actually unique and they are highly conserved. When you say highly conserved, they are not easily changed from one species to another. The ribosomes in bacteria and eukaryotes, overall, they are very similar. And still, two subunits with mRNA running between them. In eukaryotes, it's usually larger and more complex. It contains more than 80 proteins. Chloroplasts in plants and the mitochondria have also ribosomes and are simpler than those in bacteria. So the bacterial ribosome is the 70S ribosome, and the eukaryotic ribosome is the 80S ribosome. Now, the components of the bacterial ribosome is made up of the 50S and 30S, while the eukaryotic ribosome is the 60S and 40S. And my assignment for you is, what's the reason why if you add 50 plus 30, it's not 80, but it's 70? In eukaryotic chromosome, you have 60 and 40 subunit. It's made up of the entire ribosome is 80S ribosome. So what's the reason for that discrepancy? So let's move on to the characteristics of the tRNAs. The single-stranded, it is a single-stranded RNA of 73 to 93 nucleotides in both bacteria and eukaryotes. It creates, it has this configuration of a clover leaf structure in 2D, but it has a twisted L shape in 3-dimensional. Most have G or guanosine at the 5' end, and all have a CAA at the 3' end of all tRNAs. And they have modified bases. They usually have methylated bases and so on. So this is the structure of the tRNA. It has an amino acid arm, an anticodon arm, a D arm, and then the T size C arm. Okay, or the amino acid arm, okay, consists of the CCA and the CAA end. It has amino acid esterified via a carboxyl group to the 2' or the 3' of the A or the adenine of the terminal CAA codon. It should be CAA. So this portion has the CAA codon, and it's either the 2' or 3' of the end of that A or the adenine group wherein your amino acid is bound to. The anticodon arm, this is the structure of anticodon arm. It is where your tRNA will bind to the codon of the mRNA. The D arm contains dihydrouridine. It contributes to folding. The T size C arm contains pseudouridine, has bonding between base and ribose, and it also helps in folding. So this is the appearance of the D arm and the T size C arm. Okay, this is the 3-dimensional structure of the yeast tRNA containing phenylalanine. So again, it's the site of amino acid is at the 3' end. Okay, so again, what are the 5 stages of protein synthesis? We have activation of amino acids. And in this activation of amino acids, the tRNA are amino acylated. Initiation of translation. In this process, mRNA and amino acylated tRNA bind to ribosome. For elongation, cycles of amino acid tRNA binding and peptide bond formation until a stop codon is reached. And fourth, termination and ribosome cycling. The mRNA and proteins dissociate and ribosomes are cycled. For folding and post-translational processing, it is catalyzed by a variety of enzymes. Now let's discuss the individual stages of protein synthesis. The step 1 or the stage 1 is the activation of amino acid. So in this step, they have the creation of amino acid intermediate. The amino acid tRNA synthetases esterifies 20 amino acids to the corresponding tRNAs. So it is this enzyme that responsible for the binding of the amino acids to the tRNAs. The carboxyl group of amino acids attack the phosphate of ATP and it creates this amino acyl adenylate intermediate. A pyrophosphate is also cleaved, so the reaction is driven forward by 2 phosphoanhydride bond cleavages. And the fate of the amino acyl adenylates varies. So again, these are the steps of the amino acylation of the RNA. So amino acid, so it's the carboxyl group of the amino acid attacks the alpha phosphorus of the ATP, forming this 5 amino acyl adenylate. Now that 5 amino acyl adenylate can undergo this class 1 amino acyl tRNA synthetases or a class 2 amino acyl tRNA synthetases. For step 2 is the transfer of amino acyl 2 tRNAs. The amino acyl tRNA synthetases transfer the amino acyl group from the enzyme to the tRNA. The 2' hydroxyl or the 3' hydroxyl of the tRNA attacks phosphate of amino acid intermediate, creating a phosphodiester bond between the amino acid and tRNAs. Okay, so I won't be specifically dwelling on the biochemical process of the transfer because it's actually quite complex. So I just want you to note that this enzyme, amino acyl tRNA synthetase, it transfers this amino acyl adenylate to tRNA. Okay. So amino acyl tRNA synthetases. So each enzyme binds a specific amino acid and matching tRNA. So we have 20 amino acyl tRNA synthetases, one for each amino acid. Some cells have fewer than 20 synthetases, and in this case, one amino acid is converted to another one after charging the tRNA. Amino acyl tRNA synthetases must be specific for both amino acid and tRNA. The matching of each amino acid with the correct tRNA can be viewed as the second genetic code. And the code is in the molecular recognition of a specific tRNA molecule by a specific synthetase. So that amino acid tRNA synthetase, they depend on the type of RNA and the type of protein. And they are very specific. Only a few nucleotides in tRNA confer binding specificity, especially in the anticodon region. Okay. So stage 2 is the initiation. In prokaryotes, we'll discuss first about prokaryotes. The first tRNA is unique in prokaryotes. The first codon is in any peptide of prokaryotes is AUG. So AUG is the first codon and it codes for methionine. All organisms have 2 tRNAs for methionine. In bacteria, plus chloroplast and mitochondria. Chloroplasts are found in plants. Mitochondria is also found in eukaryotes. The initiation tRNA inserts an N-formyl methionine, which uses a special tRNA called the tRNA F-met. The interior methionine is inserted with a normal tRNA methionine. Okay. Eukaryotic protein begins with methionine, but not FMET. But a special tRNA is used for peptide initiation. So methionine is actually unique because it's usually the first codon. It is the first amino acid that is placed in any protein. 20 genetically encoded amino acids are common in all organisms. But 2 additional amino acids are also found in few proteins and are genetically coded. Selenocysteine are formed after charging an UGA or a stop codon, recognizing tRNA with serine in both bacteria and eukaryotes. This unique tRNA is found at low levels in the cell. Pyrrolysine is directly attached to its tRNA that recognize UAG stop codon by some archaebacteria. So these are uncommon amino acids found in some peptides. The initiation process of prokaryotes usually involves large assembly of proteins and the following requirements. So you need to have the 30S ribosomal subunit, mRNA, and the formyl methionine tRNAs, initiation factors, GTP, the 50S ribosomal subunit, GTP, and magnesium. So the step 1 for the initiation is that the 30S ribosomal subunit binds to IF1, IF2, and IF3, and the mRNA. IF stands for initiation factor. The initiation factor 3 keeps 30S and 50S subunits apart. The initiating 5' AUG codon of mRNA is guided to its correct position by the Shine-Dalgarno sequence, which is a region in mRNA that is complementary to the sequence of ribosomal RNA. The second step is the formyl methionine tRNA joins the complex. The formyl methionine tRNA binds to the peptidyl site along with initiating 5' AUG. So again, you have the Shine-Dalgarno sequence. That Shine-Dalgarno sequence is like an initiating sequence to guide the mRNA that you are already in the ribosome. Step 3 is that the 50S subunit associates. The large 50S subunit combines with the 30S subunit to form in the initiation complex. And it is the initiation factor 2 that hydrolyzes the GTP. mRNA sequences that serve as signals for initiation of protein synthesis in bacteria include the Shine-Dalgarno sequence. So in the Shine-Dalgarno sequence, these are signals for initiation of protein synthesis. And as you can see here, you have the initiation codons that is paired with the FMET tRNA. For eukaryotes, it uses more initiation factors. We have over 12, including EIFIA and EIF3, which are functional homologs of IF3 and IF1. And they have different mechanistic details and has a step that circularizes the mRNA during initiation. So I will not be dealing with the individual steps of initiation in eukaryotes because it's quite complicated. The next is the elongation step for prokaryotes. Now, I will only be focusing on how protein synthesis occurs in prokaryotes. The process of prokaryotic and eukaryotic protein synthesis, the concept is the same, but the enzymes and proteins that are involved are very complicated in eukaryotes. The first step in the elongation in prokaryotics is the binding of the incoming amino acid tRNA. The incoming amino acid tRNA binds first to an elongation factor 2 GTP complex. So this amino acyl EF2 GTP complex binds to the amino acyl site of the 70S initiation complex. So this is this green. You see this one. This is the amino acyl tRNA. That amino acyl DNA a tRNA binds to a 2-GTP complex, which is an elongation factor. Okay, and this complex binds now to the amino acid site of the initiation complex. And after GTP hydrolysis, the EF to GDP now leaves the cell. And this proceeds now the elongation. There are now 2 amino acids bound to tRNA positioned for joining. One is on the A site and the other one is on the P site. N-formylmethionine group is transferred from its tRNA in the P site to the amino acid in the A site. The reaction is catalyzed by 23S rRNA, which is a ribosome. The uncharged deacylated tRNA FMET is now in the P site. So I'll be showing you a video on how these processes usually occur. So this is the formation of the first peptide bond. So the first peptide bond starts with the formylmethionine, and the incoming second amino acid will bind to the P site. So remember, through the process I've discussed on peptide bond synthesis is the dehydration reaction. So the dehydration reaction also occurs in this step, in the peptide bond formation. Step 3 is the translocation of the ribosome. The ribosome moves one codon towards the 3' end of the mRNA, and it uses energy from GTP hydrolysis. And GTP is part of the EFG complex. It leaves A site open for new amino acid tRNA. So the process of elongation and protein synthesis, in this reference, it tells us that a ribosome is the one moving and not the mRNA that is moving along the mRNA. The elongation stop process in eukaryotics are similar to bacteria, but we have a lot of elongation factors that are present. The difference is that eukaryotic ribosomes do not have an E site, and the uncharged tRNAs are released from the P site. The termination of protein synthesis is signaled by a stop codon. It's the UAA, UAG, or UGA in the A site will trigger the formation of, or it will trigger the action of the termination factors called the releasing factors RF1, RF2, and RF3. These help to hydrolyze the terminal peptide tRNA bond, and also they release peptide and tRNA from the ribosome and causes the subunit of ribosome to dissociate so that initiation can begin again. So as long as that A site will recognize the UAA, UAG, and UGA, now causes the action of the termination factors to act, to dissociate the ribosome. Okay. So I will not be discussing about ribosome rescue and post-translational modification because these are actually quite complicated. And I think that ends my lecture on transcription and translation. It's more or less a very complicated topic because there are a lot of processes that are involved. But I just want you to note the overall process of the translation, the transcription, and replication. How are they different from one another? How are they similar to one another? Now, please focus yourself on the prokaryotic transcription and prokaryotic translation. And in terms of eukaryotes, I just want you to be familiar with how do eukaryotes undergo transcription. So for transcription, study on prokaryotes and eukaryotes. For translation, just study on the prokaryotes because it's somewhat simpler. And I don't expect you to remember all the specific transcription factors, all the specific elongation, initiation, and releasing factors because it's a very tedious way of knowing all this stuff. But I just want you to know the overall process and the steps on which these processes of the central dogma of molecular biology takes place. So thank you so much for your time and attention. and listening in this lecture on the central dogma of molecular biology. It's a very long topic, but you need to understand these concepts by heart because it will form your foundation in the further studies, especially in research in the biomedical field. So thank you so much and have a great day ahead.